The first thing I'm going to talk about really is your business model, which is also a regulatory issue for in vitro diagnostics, and even for some software types of products. We're seeing some creative business models going on. And what we have really is a split. So if you are a product that you're going to sell to laboratories, so you're a system, kind of the traditional diagnostic system, you have a instrument, you have an assay, you have software, that all comes together. You sell it to hospital laboratories, and they bring the assay up in their laboratory. Those kinds of products have always gone through FDA. So when I was a young buck regulatory person, I was taught that the reason for that was that things are crossing state lines. All right? So FDA has authority for products that cross state lines. But there's another way to get a diagnostic to the market, which is called a laboratory-developed test. And that laboratory-developed test market is usually when you're developing assays. And that's really your focus. You're not working on technology. You're not trying to make the next best sequencer or the next best PCR machine. You're really taking tools that have been developed by a lot of different manufacturers, and you're putting them together to create a unique new test. But you're not going to sell physically, you're not going to sell your PCR plates, you're not going to sell any of that. What you're going to sell in that environment is really a test result to a physician. All right, so that's the difference in paradigm. Now, some people have combined these. So we have some clients that have started out, they've been working on technology. So maybe there's one that comes to mind where they're really looking at single molecule analysis, and they developed a product that does that. They decided that they couldn't sell that right away, so they decided to create a CLIA lab around that technology. So they're kind of a different play, and later on they'll probably go back to the FDA side of things and think about going that direction. So there's some interesting ways you can use these two models to your advantage depending on what stage of development you might be in. So if we think about traditional, you go through FDA, you prove yourself in particular ways that FDA asks for. Some of those traditional models have been, Roche Diagnostics. This is mostly what Roche Diagnostics does. They develop a new PCR machine, they develop an assay on it, they take the whole kit and caboodle, and they go off to FDA. All right? But if you think about laboratory-developed test models, that's genomic health. That's CardioDX. That's a lot of models in the Silicon Valley, but also a lot of other places. They've really decided that FDA approval is not valued potentially, or it's just not something that you can get a very novel test through FDA and that FDA barrier. All right? So there's a number of reasons to think about IVD versus the LDT model. The other thing that we've been seeing too is that a lot of the software that's coming out actually gives a result. So there's lots of cool new software that takes all this data, say from a next-gen sequencer, and actually interprets something. Well, if you're going to just sell that piece of software and you're going to sell it for more than research use only, that's potentially a medical device. But what if you run that software inside of a CLIA laboratory? Could you then say that that's a service, and that might be a model that you could potentially avoid FDA with for a little while? So we see some people being pretty creative. So this has been the case for quite a while. A lot of companies have embraced both models here. So when I was at Chiron Corporation, we were developing bDNA technology, and we were developing kits, but we also had the Chiron Reference Testing Laboratory, so we could get physicians, we could get people using those we were working on viral load monitoring, we could get those results early. But our thought was always that we would eventually sell a kit, all right? And I think what's really changed in the last 10 years is that's not always how it is anymore. It's not a stage of development. It's often a place where people stop and say, we're comfortable with this. We don't need to go to FDA. Our KOLs don't think it's important, so we're going to stay right here. All right. But we've been seeing actually a third model that's been developing, and this is really around companion diagnostics. So FDA has, for this laboratory-developed test model, as Steve talked about earlier today, has shown what they call enforcement discretion. So FDA believes that they have the ability to regulate laboratory-developed tests today. So they believe that these are medical devices and that they should be regulating you. which is a different interpretation than I talked about earlier. So far, they have said that we will show enforcement discretion. We won't come after you because of this model. And the idea is there that there's a lot of smart people within that laboratory that are controlling it. All right? So, but FDA has said, so right now, FDA is in a place where they would like to regulate laboratory-developed tests. They have not been given the authority to do that yet, but they have the guidance documents all written. They would love to come in and regulate this sector of diagnostics. You know, Steve and I can debate all afternoon whether that's actually ever going to happen. Lots of people like to have that debate. So, but right now, it is hands-off. But what FDA has said is that if you are a companion diagnostic, and this is a very narrow definition, if you go to if you are working with a pharmaceutical company and you are the test that is used with their drug in that clinical trial, and they are saying that companion diagnostics need to get approved, if you are that test, enforcement discretion no longer applies. All right, so they are saying that companion diagnostics, because of the risk of that test result, that test result is going to be used to decide to give a life-saving, potentially also adverse event type of drug, something like a chemotherapy, something like that. They are saying the risk of that is too high to be covered under that laboratory developed test model. And they want to see PMAs for that. So whenever there's a new regulatory paradigm, we see people trying to figure out how they can take advantage of that. So several of our customers have set up laboratory developed tests. They are doing testing with pharmaceutical markers, or with pharmaceutical companies, on markers that are valuable to those therapeutics, but they are currently CLIA labs while they go through that process, because to give that kind of result, you would need to be in a CLIA lab setting. But they have decided, they know right now that they're going to have to go through a PMA. So they're planning, everything is eventually, they are currently laboratory developed tests to be able to service those pharmaceutical companies in those early stage trials, but they know very well that they're going to have to go for an FDA approval. All right? And FDA has found mechanisms to give clearances to those laboratory developed tests. So there's a few of them that have gone through FDA, like XDX and MammaPrint. So FDA has some idea how they want to do that. But these companies are embracing LDT with the full knowledge that they will eventually go through that FDA process. And I think this is very attractive because the pharmaceutical companies right now are bearing much of the cost for this companion diagnostic development. So instead of having to go out to those VC firms, those sorts of things, they are heavily funded by the drug makers in order to do this. And then, of course, they can sometimes develop their own passions as well. So that's something that we're seeing. It's kind of a hybrid, LDT now, FDA approval later. All right, so should you be an LDT or should you be an IVD? So if you're an LDT, it really may be appropriate if assay development is the focus of your company. So it's possible if you have a technology play that there's a way to incorporate that LDT model in there, like I was talking about with Chiron Corporation, but most likely the folks that are really focused on that assay development, this is what's going to really be attractive to them. So often it's also necessary if you have a very novel intended use. So something like genomic health or CardioDX, where you're really going into a novel place. Nobody has heard of this sort of type of diagnostic before. Often it wouldn't be enough just to be able to sell kits. You're not just doing assay development, you're really doing market development. And so that is the laboratory developed test space is often a very nice space to be able to do that in. If it is a very complicated assay, a laboratory developed test may be the best way for you to deploy that technology. Maybe really knowledgeable users that have helped you develop that test may be able to run it. I know with BDNA, it was an art form for those first 4 or 5 years, truly an art form. You know, eventually got to a place where it could be kitted, it could be run by a lot of different laboratories, but if you have an assay where 3 people are really good at this and everybody else fails, that may be something that's much better suited to a laboratory developed test environment. So it may be something that depending on what kind of techniques you're using, it may be a way to get out there a little earlier than maybe the kind of robustness that you would need for a kit type of test. The other thing that's very necessary is you need to find an intended use for that product that is okay for the physician to receive that result in 3 to 7 days. So a couple weeks ago, I was meeting with some folks that have a new technology, but they're really interested in sepsis. And so that doctor needs a call on sepsis now. It's not going to be okay to take that sample, put it in FedEx, hope it gets to the lab, have the lab do their extraction, turn it around, and great, the patient's dead, but you got a guaranteed correct result on sepsis. That's not something that's going to do well in the laboratory developed test environment. That's something that you really need to have a kit for, have it be at the hospital laboratories, and be able to get that kind of test result right away. So that's the other thing to look at is a test result back to a physician in 3, 4, 5 days going to be acceptable? And if 3 days really is the longest you can go, that's really on the edge, because guess what? FedEx makes mistakes, instruments in the lab go down, so you really need some play in that timing in order to make that laboratory developed test model work. So now the other thing too is sample stability. So the other thing when you're going to send a sample through FedEx, something like that, you have to make sure it can show up in a day or two and still be okay to test. All right, so if you want to go down this pathway, and there are some things you can do, there are interesting stabilization technologies out there that could facilitate this more, but that's something you need to look at as well. The other thing is if your key opinion leaders don't value FDA, that's great. Believe it or not, there are a few, and Steve might argue with me on this one, there are a few areas where people actually value FDA approval. So we'll get into that next. So if you're developing an IVD, so again, if you're going through systems development, that is likely going to be something that you're looking at selling hardware and reagents as your revenue model. All right? So if that's the revenue model you're thinking of, then you're likely going to need to go through FDA, because you're going to have to ship that stuff to a lot of different laboratories, and you're going to demonstrate that a lab in Chicago can do it, and a lab in Arkansas can do it, and a lab in Texas can do it. And that's going to be the expectation from FDA. So the other thing too is remember your customer is very different. So with that laboratory developed test, you're really selling that test result to physicians. That's your market. If you mostly like to talk to labs and find out what they want, then that's the market you want to develop with an in vitro diagnostic. All right? So again, I think I already mentioned that the assay results are needed quickly, and then the assay is point of care. All right, so I went to the Next Gen Sequencing Conference a couple of weeks ago, and there was these great pictures of these, we're going to bring Next Gen Sequencing next to the patient. And there seemed to be some sense that that could all happen in a research sort of way. And that kind of device, something you're going to put in that physician hand, needs to go through FDA clearance or approval. I really don't see a way around that. You're also going to have to do some other things, but we can talk about that another day. So again, the sample can be shipped, so you want to do that. And then if your KOLs truly expect FDA approval, and your KOLs may be your payers too, if they truly expect FDA approval on that, then this is the pathway you're going to want to go down. So clinical utility is extremely important. This was talked about in the reimbursement area this morning. And I think sometimes people decide to go down that laboratory developed test pathway because to physically get on the market, to be able to offer a product, there's the folks that regulate CLIA, CMS, they really don't look at scientific evidence. They don't. You can, as long as you can show, let's say I decide I have a new marker for acute kidney disease. And I really believe that that marker can do some good. In a CLIA lab environment, I don't have to have all of that data to start offering the test for that marker. As long as I can show I can do that test reliably, I can offer it in a CLIA lab setting. But to go through FDA, if I come to FDA and I say, I have this great new marker, and I don't want to tell you why I want to test it, I'm just going to tell you I'm going to test it, FDA is going to say, sorry, I can't clear something like that. So the intended use for that needs to be told to FDA before they can evaluate whether that test is functioning properly. So it's another level of understanding what a marker has to do to be able to get through FDA. All right, so, but I think what most people have found is that when they set up LDTs and they're like, cool, we're on the market, we can avoid FDA, is their next leap is the payer. So you may be able to get some early testing done, you may be able to find a stacked coding kind of way to go, but eventually, if you're successful, somebody's gonna ask you for the data and why you think testing that marker is important. All right, so just no matter what you decide in terms of laboratory developed test or in vitro diagnostic, I strongly encourage you to consider the clinical utility of the marker. So, and that's a hard bar. That can be a very difficult bar to reach. So the focus really needs to be on those studies. Of course, it's interesting to develop those assays, but you're gonna have to take the next leap and decide really what they're good for. So we hear a lot in diagnostics about all of these amazing cohorts that are out here. So probably the thing that makes my head hurt the most is when somebody comes to me, they have an early technology, and in that business plan, it says, we're gonna test 500 samples. And it appears to be a mystery where those 500 samples come from. So I had a person in my office last week who's developing a novel new test in women's health area, and in his business plan it said, and we'll test 500 samples before we get on the CLIA marketplace, or before we offer our test. And I said, those look like magic. Where did those 500 samples come from? And he said, well, we know all these docs at Stanford and all these great places, and they all have stuff in their refrigerators, and they're gonna give them to us because they trust us. So it's all gonna be good. We're gonna have these magic 500 samples. Or right now, I'm also working with somebody that is getting a product through. It's gonna be an FDA product, but they decided that they need outcome study. So it's a risk sort of test. And so they bought a great cohort. So it's a big multi-study, multi-state study in this particular disease area, and they bought this cohort, and they tested it, and they sent it off to FDA, and they said, huh, that's great, huh? Look at this, we have outcome data. And FDA came back and said, and this was before I got involved, said, the population on that study is not the same as the intended use of the population that you're going for with that test. Thank you, but no thank you. So now we're having to go back and see if we can justify that as a cohort. All right, so when you think about your test, think about what the paradigm is you're going to be using it, and think about whether it's worth doing a prospective study. So as far as clinical utility, my suggestion is, and I'm always a little surprised when people get to the point of considering regulatory when they haven't had this suggestion. Go to whatever the biggest whiteboard you have and write down what the diagnostic paradigm is today. And I mean everything. So if you're going into neurology, write down what kind of assessments do the physicians make just on visual with a patient? What do they make based on questionnaires? What do they make based on imaging? What are all of those pieces of the diagnostic puzzle? So write that down, go talk to people, find out what that actually is. It may vary by institution, so it may not be one answer. And then start your flowchart with how your test is going to fit into that. And you'll be guessing. You'll be guessing initially. It will change as you've developed your test. But this is really, so a lot of people come to me and they say, we got this great new test and it's in, it's for assessing whether people have broken legs, and it's multivariate, and it's really cool, and what do you think FDA? What's the FDA going to say? Is it going to be class 1, class 2? What's it going to be? And I have to make them go through this paradigm to see, are they going to replace x-rays? Not that you could do this with biomarkers, but people like to try all kinds of stuff. But are you going to replace with that test the existing paradigm for diagnosing of that disease or condition? If that's the case, then FDA is going to want more scrutiny on it. We're just going to stop using x-rays, and we're going to use your biomarker, and we're going to get all the broken legs that could ever be. That's going to be a higher bar than if you're going to say, actually, you should still use an x-ray, but my test will tell you if you should use X kind of cast or Y kind of cast. That's a different level of risk. But you really need to think about what that's going to be before you can talk to FDA and before you can design that study. So, and actually these flowcharts are exactly what we put into documents when we go talk to the FDA. Because you're talking to a group of people, much like payers, who may not know that disease space even close to as well as you do. So it's really important to write this down. It's also important to test your assumptions. So, so coming back to the cohorts, so there are some challenges with using those existing cohorts. Like I said, the samples were collected under a protocol that was not designed to test your diagnostic. All right, and we see this also with drug protocols. So a lot of people want to say, we have this great new diagnostic, it's going to tell you risk of a certain kind of cancer, say, over time, and we've got this cool drug protocol, this cool set of specimens from this drug protocol, this therapeutic protocol. It's possible that the folks in that protocol are not actually the folks that your test is going to be targeted to. And FDA is going to worry quite a bit about that. So the other thing FDA asks for, and this is very difficult to overcome, and this is particularly true with protein biomarkers, is what is the stability of that marker over time? So if you're working with a cohort that has been stored for 10 years, and you just want everybody to accept that that hasn't changed, and the result that would have been gotten at time 0 with that sample is the same as the result you get 10 years later, when it's been stored, maybe frozen and thawed a few times, FDA is going to ask you for evidence for that. Now that can be an absolute showstopper. Because how would you ever show that? If you're now measuring a new protein that we didn't know about 10 years ago, how do you ever show that that was stable over 10 years? That's the impossible challenge. And I've seen some pretty creative scientists come up with some thoughts there, but it can be very, very difficult. So the other thing I see is people get these cohorts and they say, okay, we listened to you, Maya, it's the same population. And they've really done that well, but their test is designed for plasma, and guess what? That sample, it has been frozen or it's for EDTA, but that whole cohort has been stored in heparin, something like that. The sample type is slightly different than what they want to go to market for. So is it then the same assay? Is that appropriate cohort? So we've seen a lot of issues with that over time. So for the other thing we see is, I have one client that they're developing a risk test, and they started on this a number of years ago. And the first thing I said to them is, okay, let's get our sample collection sites set up. You're going to need a prospective trial for this because it was a risk over time type of product. And they said, no, we don't have time for that. We're going to be on the market within a year. Forget it, Maya. We're not doing prospective studies. I said, okay, well, let's start looking for the cohorts then. And we had all these committee approvals, and some of the cohorts only had 50% of the samples left. And so lots of contortions and trying to figure this out. 5 years later, they're still trying to get those samples. And it kills me, and it's hard for me not to say I told you so, but I'm like, we had 5 years to collect these samples, people. Come on, let's think about this. And even if you do have that cohort, what are the chances are, so let's say you're super lucky, and let's say it's maybe even your cohort. Maybe you were an investigator at a university, and you have that cohort yourself, and that's part of what you're starting the company on. What are the chances down the line that a payer is going to ask you for a second study at some point? That's pretty highly likely. So really think about that. And often the cost and time for obtaining those samples, it's much more than you might think. So I think I've made this point already. Seriously, please, even if you're in that diagnostic space, it's taken for granted with therapeutics, please consider a prospective clinical trial. Make sure you have an endpoint, make sure you're collecting those, make sure those are your property. I can't tell you how valuable that is and how much more successful the companies I work with that do this are than the folks that try to skip this piece of the development process. So the other important thing to understand, and I didn't put this in the slides, is FDA expects you to develop your test on one set of samples and to verify that it's working. They expect that final trial where you really come up with your performance characteristics, whether that's multi-lab because you're IVD or you're an LDT, on a new set of samples. You cannot develop and verify your test on the same set of samples that you validate your test on. All right? And it's not just FDA that thinks that. A lot of people will say you are cheating if you use those development samples for validation. I've seen some pretty creative statisticians try to convince me of this. I asked this question once and only once at FDA, and I used to be 2 inches taller, actually. They just about took my head off to even suggest this. All right, so payers think this is important, FDA thinks this is important. Assume you are going to need 2 sets of samples in order to get all the way through. The other thing, too, is I have seen occasions when people are not successful, and the only asset they end up with is that set of samples. And if it's in the right population and it is a good set of samples that somebody else could use for verification, that may be the asset that you have. All right, so it's not just money out the door. Consider it an asset and consider that you're building that as part of your diagnostic company. All right, so if you do decide to go to FDA, so even if you're that LDT model and you do eventually decide to go to FDA for payer reasons, because you want to do something different, FDA is always going to be concerned about a banked cohort. I've never, ever just gotten through that discussion with FDA in an easy way. And they're very concerned about bias as well as a number of things. So if you do see FDA on your horizon, or you're one of the believers that says FDA is going to regulate these tests, really do consider a prospective trial. So I'm just going to go over a couple potholes, and then we'll start the panel discussion. So software. Software is often a medical device. Often is. And FDA, believe it or not, there's a few people at FDA where it's their job just to troll around websites and see when you're marketing your product inappropriately. All right, so if you have software, it does make an interpretation, it does make a clinical determination. It is possible that you're a medical device, and you may want to be very careful of your claims before you have the chance to go through FDA. Specimen collection products. So this is one where folks have gotten in trouble, and good old Pathway Genomics made this public news when they decided to sell a saliva collection device at Walgreens for their direct-to-consumer assay. A specimen collection device is a medical device. Best case, it's class 1, likely it's class 2. So this is something that is not covered by enforcement discretion around LDTs. All right, so if you have a novel stabilization product, something like that, that needs to be in that tube, and you want to sell that, and that's going to be the front end of your LDT, that is a medical device. All right, and there has been a saliva collection device that has been cleared by FDA. So a buccal swab, if it makes claims for stabilization for DNA or RNA, is actually a class 2 medical device. So, and there's been a number of companies that have gotten in a bit of trouble with this one. So supply chain, I put this up here because FDA, they haven't gotten authority to regulate LDTs directly, and some would say they're attempting to do this indirectly by messing with the supply chain that goes into laboratory-developed tests. So the majority of laboratory-developed tests rely on the heavy reliance on the use of products that are marketed as research use only. FDA has put down a draft guidance document that says it is inappropriate for manufacturers to sell those products if we know that they are going into clinical diagnostics. All right, now how they're going to enforce this, whether they can actually ask us to enforce this as manufacturers, are all good questions that are on the table. But FDA has enforced this in the past periodically. The most recent public warning letter was to Illumina when they were selling research use only arrays to 23andMe for that direct-to-consumer testing. They knew very well those arrays were being used for a clinical diagnostic test. It was in a CLIA laboratory, and FDA said, not okay. That's unacceptable. So if you've decided to go LDT, probably 80% or more of those materials that you're using are research use only. And if FDA does get the authority to go after this, that could be a significant problem for you in the supply chain just to get the products that you need. All right, so this is a little bit, if you can't beat them on the front lines, starve them out on food supply kind of technique here. But it is something of concern to a lot of the laboratory-developed tests.
Are there databases where
There are. So there's definitely, you can find out what's cleared. You can find out who's registered with the FDA. The challenge for a lot of reagent manufacturers as well as instrument manufacturers is FDA has a policy of systems that have to come into FDA. So in order to get, say, a PCR machine cleared, I can't just take that PCR machine to FDA. They want to see an assay on it. All right, so which is part of the reason most of the PCR and sequencing, even Sanger sequencing instruments are still RUO. And you've got the same problem on the reagent side. So I may make a very unique type of reagent, but it doesn't qualify as a general purpose reagent, which is a nice little category for some reagents. There's really no way to do anything but to sell that as a research use only. And that's, I send comments into FDA about the research use only policy, and that's my biggest gripe with this, is it's either nothing or everything with you guys. A lot of us want to do the right things out here. There's just not a regulatory way to do that. And I give them a hard time about that as much as I can. So you will have other stakeholders too. So if you are a CLIA laboratory, many of the states will not do much more than CLIA, but if you want to sell that test in the state of New York, you've got a very high bar that includes clinical utility that you're going to have to go for, which is why often LDTs do limited launches. It's rare that you do an LDT and launch in all 50 states for a lot of reasons, but part of it is regulatory in that state of New York is pretty tough on these things. And state of New York sometimes doesn't let it in. So state of New York has still said, we don't want direct-to-consumer genetic tests in our state, thank you very much. We don't see that the science there is enough, we don't want you here. So they are allowed to say that. There are, of course, other countries. Believe it or not, there are people outside of the US, and sometimes your model's going to change depending on who you want to serve. Often doing a laboratory-developed test, it's very difficult to do something where you have to ship something from the other side of the world. All right, that may just not be a possibility. So you need to think about what you're doing, and then, of course, the payers. So a lot of people like to go to the EU first. For LDTs, it's a bit vague. But there are quite a few LDTs in the United States that do accept samples from other countries. There are ways to do that. So that may be something you want to consider. For in vitro diagnostics, it is definitely a lower bar to go to the EU first, and you typically do see people marketing there prior to marketing in the US. So it's just something to consider as you develop your plans. And I think it's time for the panelists to come up.
So I'm Steve Rosenberg. I'm the CSO of CardioDX. I've actually been lucky enough to be involved in the development of 3, what were originally LDTs, one of which is FDA cleared, at Genomic Health XDX, and now CardioDX. And I have sort of responsibility for both research and development. Research involves everything from sort of biostatistics to some clinical development when we didn't have a CMO, through a lot of the laboratory and also things like next generation sequencing, which we're dabbling in. And get to go to FDA on occasion with Maya to protect me. I think that's probably all I want to say.
I'm Una Ryan. I'm the CEO of Diagnostics for All. I met many of you yesterday, I think. I'm very fortunate that 2 companies ago I ran a public vaccines company, and regulation for vaccines is regulation on steroids. Yeah. So afterwards, diagnostics doesn't really seem easy, but it's just not as bad. And the manufacturing requirements are not nearly as bad. So I tend actually to be over-compliant on a lot of things. But those of you who were here yesterday recognize that I tend to do things backwards as well. So we have a nonprofit that owns its for-profit. And I've done this before in vaccines. I tend to think that it's better to launch outside the US first and come back to the US last, which is quite opposite from the way you learn things in business school, which is half the world's market is the US, and then there's Europe and the rest of the world. We think the attractive opportunities are in the emerging markets and in the developing world. So we've designed our approval processes to let us go backwards, essentially.
Okay, I'll ask I'll ask the first question. Rena, I'll start with you. Related to you, your overall mission is really to get great products for high unmet needs in the developing world, and that you may have the opportunity sometimes to take some shortcuts or shorter cuts. How do you balance that off, or do you make any trade-offs in terms of speed to market or speed to getting those products in the hands of patients versus meeting regulatory requirements of some countries that may be more stringent?
I think that's a very important question, and I really want to make a very strong ethical answer. I remember various governments saying this to me about the rotavirus vaccine we were developing. If it's not good enough for American children, it's not good enough for us. Don't come to us. So I feel very, very strongly ethically that you don't dump things on developing countries that you wouldn't be willing to sell in the US. But having said that, I think that the benefit-risk ratios are often different in the developing world, and you can make the case where children are dying of a disease that you really need a diagnostic, a drug, or a vaccine much more easily than you can in the US, where you have to, well, you're up against parents missing a few days of work or something. So I would never say that we do it for shortcuts or to sell shoddy products. But I'll just tell you, my original thought was if we're going to the developing world first, we'll go for WHO pre-qualification. And then all my friends from the larger companies said, oh, it's backed up for 2 years, it's just awful. You didn't have to have friends. You could read every day in the paper the problems with the FDA and the 510K process and the lack of clear instructions. You know, we have guidelines, but we don't really have a checklist of things we have to do. And I will say to Steve that the one or the areas where I've found there is appreciation of the FDA is overseas. You know, we had some potential partners in Brazil where we were going to go into primary clinics in Brazil. And I said, oh, wonderful. You know, we don't know the regulatory process in Brazil. You can do it. And, you know, you probably went to nursery school with all the ministers and things. They said, oh, no, we want you to get FDA approval. We'll send Brazilian inspectors to your manufacturing. It's just a room in Cambridge. I don't want it to sound like a facility. You know, I was actually bowled over that they wanted that. But the upshot is that what FDA will do with it, what DFA will do with its own money is we will get our products first approved with CE mark and, you know, get our manufacturing approved through the European system, and then with partners who want for-profit uses in the US or find ways to get use of our product, our technology without, and I can give some examples of that. That's how we will introduce things in the US. Sorry, it was a long answer.
A great one, though. My next question goes to Steve, and it's going to bring you back to your earlier discussion today. So
Okay.
when you once you've sat down and really have a healthy appreciation of the different trade-offs that are available to you in getting your product to market from a regulatory standpoint, but also now understand what are some of the elements you may need to build up as part of a reimbursement pathway, how do you balance the two in terms of choosing what you do or not do, in terms of both costs, timelines, and access to patients?
Well, that's a good one. So I've been living that one for 10 years. So I think at the end of the day, a lot of it is a question of access to capital. I mean, to Maya's comments, if you can do the prospective studies and they turn out as you would hope they would, no one's going to argue you've checked the box. It's done. The challenge is the following. So a number of the tests that I've been involved with are diagnostic tests in the sense of what you want to know is what is going on with a patient today. You're not looking for prognosis, what's going to happen in the future. You're saying, patient has a certain set of symptoms, I want to do a differential diagnosis. Is it disease A or disease B? Or disease A or not disease A? You may not know whether it's disease B, C, D, E, F. The advantage of a diagnostic test is that you have a very short-term, usually gold standard endpoint you can compare to. In the case of the CardioDX test, Course CAD, we did the development and validation in 2 separate cohorts from one trial where we had invasive angiography and the picture of what the person's heart looked like 6 hours after the blood sample was drawn. And we then went and took it to a core laboratory to make sure it was done as rigorously as possible. But the endpoint was available almost in real time. That's a big advantage because that means when the last patient into your trial, your trial's done, right? As opposed to a prognostic or outcomes trial where the last patient in is almost the beginning of the trial because then you have to wait. You're waiting for outcomes, okay? In the latter case, it's interesting. If you can't use retrospective sample cohorts, it's extremely expensive to do. So we're lucky enough now, we're negotiating to be involved with a large prospective National Heart, Lung, Blood Institute study that will enable us to look at the prognostic ability of our test. It's a $100 million study. Maybe we have $100 million overall over the last 7 years into CardioDX, which is a lot of money to put into a diagnostics company. There aren't very many, and there probably won't be again, to have that amount of resources put in. So, but for example, in the oncology space, there are large numbers of retrospective cohorts, especially if you're doing something from a biopsy specimen, where you have paraffin-embedded sections from all of the cooperative groups that run the big oncology trials. They have thousands and thousands of samples with very good clinical trial pedigrees there. However, you look at Genomic Health, which is a great example. Genomic Health has never done a prospective study. All of their data, their validity data, is based upon retrospective cohorts. Now, the NCI is actually running a prospective trial today that Oncotype DX is part of, but it's not really to show validity. It's actually to ask a more sophisticated question. But they have been able to get acceptance broadly and are really clearly the market leader without ever having done that. But oncology is perhaps a special case. We have run 2 prospective clinical validity studies, Predict and Compass. The first one was really not in the intended use population. We knew it wasn't the intended use population. We went to FDA and told them we were doing it not in the intended use population. And they said, yes, you have to do that because that's the gold standard. Then you have to do the prospective study in the intended use population to show that the performance characteristics in the population it's really to be used in clinically are, in fact, correct, and what are they? So that's sort of, I think, the way you need to do this. But it's time. I mean, we started working on this 6 years ago. We launched the test 3 years ago as an LDT. And then, even post-launch, then did a number of studies, continue to do studies, continue to build the evidence base, and expect we will get significant reimbursement 3 years after launch, hence that second value of death I was talking about earlier, which is we've been eating the cost of the 28,000 tests we've run to date because most of them are not getting paid for. So, interesting business model, as some of my board members say.
I'm going to ask one last question, and then I'll open up to the audience. This is an open-ended questions for either of you to answer, or you three of you to answer. It's trying to do a little role-playing game. Imagine you're a postdoc graduate student, and you're either an inventor or are not aware of some really exciting technology you'd like to develop into a diagnostic. And you've just heard from you that it's quite complicated, a lot of moving parts. How would you encourage them to think about a series of to-do? How would you sequence your thinking or prioritize what to think about first as it relates to this important topic of clinical regulatory strategy?
Well, I would cheat the question a little bit. which is to say I wouldn't develop the technology first, then go around looking how to get it approved. I would take Maya's beautiful slides, read them carefully, and I know that stuff, and then say, this is ridiculous. This is a terrible way to get a product on the market. Let's invent some technology that, and you don't do it to circumvent the US rules. But, I mean, Americans are so impatient normally. They want results immediately. You would allow 6 days. In the developing world, if a mother has walked 4 days from the bush with a baby to a clinic, she's not going to come back next week. You've got to give a result immediately. So our tests are point of care. They're real time. Our funding is real time. There's nothing kind of in the bank. So we collect. You get a result in minutes. And we're very careful that you can read the results immediately on the chip. You don't need any equipment, anything. But if, it has a shelf life of 2 years, but in 15 minutes, it's gone. These are use and dispose. You put a match to it. So if you go off to lunch, you've lost our test. But if you want to record it, you can take the picture with a camera phone. But we go to enormous lengths to be absolutely clear. You don't need it. There's no algorithm in the phone that you need. You can have one that says where the nearest hospital is. And our partner, Qualcomm, has absolutely said there is no way any of our software is ever going to go near the FDA, and, we're going to be your partner. Don't take us there. So we live under very, very tight restrictions. I mean, we have a milk test that has to be done in less than a minute and costs less than 5 cents, because it has to be instantaneous, because otherwise they have to put another person onto the milk line. When you live under those requirements to have something that's useful, the FDA seems easy, almost, because we simply don't have shipping issues. These are like postage stamps. You can stick 1,000 in an envelope. It's extremely easy. But there is one thing that Maya raised that I want to raise, and that is that sometimes you do need a permanent record. We were talking to a life insurance company that wanted to do a profile of risk of people. And they wanted 20 tests. We multiplex, so they wanted 20 tests on something the size of a credit card. You take that to the FDA, you've got to get every test approved. It looks like one test, but it's the same nightmare as having 20 single ones. And they wanted something for legal reasons that they could come back and interrogate years afterwards if people sued them for not getting life insurance or something. So what we have done for some of our big pharma partners is come up with our test, which allows them to do clinical trials with people at home much more cheaply. But you can put a dried blood spot on there too, which is really just the back of our tests, which filters the blood out anyway, so it doesn't mess with the color change. So you can find ways to do both, but often just putting everything on one chip doesn't actually make the regulatory pathway easier. It makes it easier for the user, but back to my Dolly Parton joke, it costs a lot to get that cheap.
Yeah, and multiplexing is something that FDA is really, they have cleared some multiplex tests. They have looked at microarrays and managed to clear those. So they have some regulations, some ideas around this. That multiplex is like next generation sequencing is something FDA is proactively going out, trying to work with the manufacturers to figure out how they can get that into the system and get that regulated in a reasonable way, and they've had some public meetings on that. So the multiplexing issue is a big one if the FDA, even if FDA figures out how to do it, you do have to show, we're working with a product right now, it's going to be in the blood screening area, and there are 37 analytes that are on that product. And the manufacturer, and some of them are difficult to find, some of the rare alleles. And the manufacturer is like, well, FDA is just going to have to understand that we can't test those alleles. And I understand that frustration, and I have to say, they're never going to buy that,
Yes.
guys. You know, and that's been a tough pushback, because to get those rare alleles, they may have to go out and test 1,000 samples to find the ones that they want. But that is the expectation at FDA. So we try to argue, we try to do as much as we can with the FDA. We try to get them to be as creative as possible. But if you are looking at a multiplex environment, it is going to be a challenging space to try and get through FDA.
But on the other hand, you can be saving lives in southern Sudan for pregnant women without going through the FDA.
So if you come back to the developed world for a minute, the first thing I would tell to your person is, go find some people who might be customers for this and find out if they would care whether you develop this or not. Because technology now is ubiquitous and really enables you to do many different things. But what you have to look at is, what is the current state of the art of how differential diagnosis, because that's what diagnostics do, works today in the environment. And it also depends who the provider is. Is it a hospital? Is it a primary care physician? Is it in southern Sudan? Where is it? What is the current way in which the decisions are made? And then say, okay, if I had something that had the following characteristics, would you use it? What do the characteristics need to be? Turnaround time, sensitivity, specificity, et cetera, et cetera. And how much are you willing to pay for it? And all those things are absolutely relevant to not whether you can develop the test, because you probably can, but whether it will ever be used and will ever make a difference, and whether you'll ever get paid for it, which is a nice thing to have happen too eventually.
And the one thing I would encourage is that, I saw on the Canvas plans yesterday that FDA was often listed, sometimes in key partnerships or sometimes in, I don't want to do this, but I would suggest that you put FDA into your customer bucket. So they are not people that are going to actually purchase your test, but if you don't think you have to sell FDA on your technology, if that's the route you're going, you're wrong. So, and part of what the sensitivity specificity is going to need to be, FDA may have some pretty strong ideas on that. So the one thing I was seeing yesterday that I'd really like to everybody to think about is put FDA in your customer bucket, because they're going to be a pretty important point if that's what you need to do to get that product on the market.
Thank you. So we're going to open up for questions. I don't know if we have a microphone that's portable. If not, I'll ask people to be loud and we'll just repeat your question for the benefit of the taping and the audience.
Okay, your presentation is impressive.
Thank you.
And I have a question regarding to the royalty. And in the last 6 years, I'm lucky to get funding by CDC. We recruit 50K patient specimen. And also, I have patient sample from Europe, which is I have MTA. My question is, if I have profit, should I pay royalty to that? 1. 2, my assay is developed by existing platform, commercial platform. Should I pay the vendor royalty?
Please repeat the question for the benefit of the audience.
So I think the question is, you've gotten specimens both from CDC as well as other parts of the world.
Yeah.
When you're a successful diagnostic, do you need to pay either CDC or the people that donated that royalties?
Actually, I go through all of the patient samples. And now I'm run commercial sample collected in America.
Yeah.
So when I get profit, should I pay the royalty to CDC? Definitely no. For the Europe partner, I have MTA. Should I pay to that?
It's a business question.
Yeah, it really is a business
depends
question.
on your contract.
I mean, from a regulatory perspective, when you've collected that sample, you would have had to get informed consent. And typically now, informed consent forms do say, by giving this sample, you're basically releasing your rights to any technology that's developed from that. So when that's pretty common, I sit on an IRB, and I see that in almost all the informed consent forms that I review. So if that's the case and you've gotten appropriate informed consent, then I would say you don't necessarily owe anything to the people that have donated those specimens. That said, I've done a lot of work in influenza, and to get difficult specimens in influenza, often we want to go to different places in the world. And some of those places in the world have been saying no, because we take their specimens and we develop great diagnostics for the US, and we never return that value back to those countries for a lot of different reasons. And so This has been a point of sensitivity for a number of countries, and in particularly certain diseases where they never actually see the value of that diagnostic work that's been done. But I would say if you've collected them appropriately, then you shouldn't owe them royalties. The instrument manufacturer is probably a different situation, and there it really depends on, I think, your business model. So if you want to go through FDA, you are likely going to have to convince the maker of that instrument to go through FDA with you. If that's the case, they're going to want a lot more than royalties. Or they may see it as an opportunity if they want to go through FDA anyway. If you're an LDT, you may have a different situation. But I don't know if you have any other thoughts, Steve.
Yeah, I think the informed consent piece you mentioned is really key. I mean, one of the things, if you're collecting samples, if you don't have the right informed consent, you can't use them. And this is, a number of people have gotten bitten by this one because the awareness of using especially genetic information, which is now covered under GINA, a law that was passed last year. But even other, you know, if you go back to how many of you read the Henrietta Lacks book about HeLa cells, I mean, there was no concept at that point about, well, if you took cells from someone that person had any rights to them. You were a patient, doctors could do whatever they wanted with them, that was that. Well, that world no longer exists. And so you have to be very careful in terms of when you set up, I mean, one of the big issues with running your own trial, and even accessing retrospective samples, maybe even more so, is read the informed consent. Make sure you're allowed to use those samples for other than what the intended use was for the trial that was run. That's really key.
A really good point. I'm going to add that actually to my slide, because certainly in oncology we've had this situation happen where when we've gone back, that's not in the informed consent form, and so what looked like a beautiful cohort with outcome studies or with outcome data,
Can't use.
couldn't use it. Couldn't use it.
But I think there's a huge lesson for entrepreneurs here, which is to get it worked out in advance, because it isn't simple. I mean, we did the US studies at the Beth Israel Hospital, and the investigator said, oh, we don't want to be paid, we just want authorship. And the PI wanted lead authorship. But we had to get the Beth Israel Hospital to sign and agree that no money would subsequently be asked for. And again, with some very big trials we've been doing in Vietnam, it's just been a nightmare getting the permissions from the AIDS clinic, the drug manufacturers, the NIH, PATH, the Gates Foundation, all these people. You really have to work it out in advance.
Well, that's the other thing I'll say. I think it came up a little bit this morning is if you do see FDA as your eventual pathway, you do have to file financial disclosure forms. So be very careful how you're compensating some of those early investigators. If they end up being the investigators on that trial, you will have to tell FDA that they are investors and that they do benefit if the trial turns out well. So that is always a concern of any of the regulators.
Yeah. I would add one last point to that so we can get to another question, which is it really pays, if you can do it, to get whoever is the principal investigator in your trial not to be compensated in any way.
Yes, that's what we did.
And the same thing with authorship. Even though having them as a senior author, where they have the credibility of, I'm not getting paid for this, I do this because I think it's good science and will lead to improvements in healthcare, is really key. It's not always easy to do.
No.
So what's the barrier with the FDA to file a 510K, to file a 10K versus a PMA?
You were right the first time, and it's 510K, which I always get mixed up when I have to talk about my 401K plan. I always call it a 510K plan, but which is opposite. Most people come to me and they say, what does it take to file a 401K? But no idea. So it really just, so the big answer is it depends. So typically for 510Ks, for in vitro diagnostics, you are having to do some kind of a trial. So with a lot of medical devices, it can be simply a technological comparison. The in vitro diagnostic world moved away from that. They started doing that at least 10 years ago, and it's rare that we have a device where, it's never that we have a device where we don't have to go out and do the studies. So for a 510K, usually it's a known marker. You can either compare it to another test that's on the market. So let's say you had the next best CD4 test. You could pick one of somebody else's CD4 test and show that you have the same results. So sometimes that's available. More often, we're looking at markers. If we're in genotyping, FDA is expecting us to compare to bidirectional sequencing. If we are in expression, then more often we have to find an endpoint like Steve was talking about, or we have outcome data. But it depends on what the marker is and what the intended use is. The big difference between a 510K and in vitro diagnostics is not so much your clinical trial anymore. It's really how much information you have to submit to them about how you manufacture and how you develop that product. So we just recently sent in a module for a PMA for one of our clients, and it took up, if we laid all the volumes out, they would have stretched across this table. So it is quite a bit more. One way I try to put this in perspective for folks is the user fee around a 510K, the medical device user fee, is about $2,500. The user fee for a PMA is about $250,000. So that's the difference FDA feels it takes in resources to do a 510K or a PMA. So the number that Alan May put up yesterday, that there's only been 10 PMAs, and I think he's off by one, I think there might be one more, is because of the size of that application. And in diagnostics, you can see from that laboratory developed test, we're kind of allergic to regulatory at all. So there's not very many diagnostic companies that really want to go after that PMA. It's mostly Roche and Abbott and a few brave souls other than that. So, and the challenge too with FDA is, let's say you did go for a PMA, you have to make sure it's not something that's easily copied by a laboratory developed test. Because you could do all the things FDA wants you to do. You could go through that application. If it is a single marker kind of product, right now there's no protection for you that a laboratory developed test couldn't develop a test for that same marker and go ahead and undercut your prices and your time to market significantly. So this is a huge problem right now. So if you look at the PMA, there's a PMA for a BRAF test that Roche did. There's lots of laboratory developed tests that have BRAF tests too, and those are currently not being shut down. So I think it's a little bit like the compounding issue in therapeutics that was brought up this morning. Sometimes even if you go and do that FDA approval, it doesn't protect you from the pharmacy compounders.
I think the other aspect, Maya, you may want to comment on this, is the level of risk that FDA assigns to the decision you're trying to make. That's sort of where the FDA will tell you, this is a 510K, perhaps it's a de novo 510K, which means there's no predicate assay that you can compare to, or it's a PMA. And I think if you're talking about, I'm going to give you this chemotherapy drug versus that chemotherapy drug, if you have breast cancer, and if you are wrong, the person is likely not going to survive as long. FDA will look at that. It probably is a PMA, I would imagine. Whereas if you're using your test in addition with a lot of other information that the physician has to help make a decision, they tend to look a little bit more towards the 510K end of the spectrum on these things.
And FDA really does want things to go to 510K. They don't enjoy reviewing necessarily the giant applications any more than we enjoy putting them together. So they do tend to be creative with us in trying to go that direction. But it is significantly more to do a PMA.
With the preventive healthcare business, and we have an assay that we have a potential customer that's really large, and they're potentially willing to use our assay, but the assay has not been tested for a clear outcome because it takes about 15 years to link this assay to the outcome. Since it's preventive, it's not diagnostic per se. So if your customer is willing to go with you on a 15-year sampling, does it require us to have FDA approval?
Not necessarily.
Sounds like it could be an LDT,
Yeah.
assuming that you're running it in the same laboratory you
That's
developed
right.
it in.
Yeah.
We run everything.
Yeah, so then if you comply with CLIA, you should be okay, but I would definitely send that customer a lot of chocolate. That's quite an investment they're making. So yeah, but to test any specimen in the United States and you're going to give a clinical result back, the very minimum is CLIA. So.
So I make software for molecular biology research services, and with the increasing amount of translational research combined with review boards, multidisciplinary care teams, things like that, I'm seeing a lot more
Please paraphrase the question.
Sure. So the question is, we're making software. Some of it's highly complex. We're taking probably a lot of different variables, putting it in the software. Often, it's potential that a bioinformatics person would actually be part of the care team to utilize this software. And is that software a medical device? So the answer is probably yes, but it really depends on the claims you make for that software, and it depends on what that software ultimately gives out. Is it basically a database that stores all that information, but it doesn't have any algorithms built into it that would say, give drug A or drug B or do this or that? Then you may be a very low classification medical device and you just need to register. But if you want to be out there making claims that this will take all of your next-gen sequencing data and tell you whether this person has a disease, then it would definitely be a medical device. So, and there have been cases, there was a big warning letter on this, it's a little while ago, but a company called Choreologic had developed a product that they were selling to LabCorp that was a ovarian cancer test. And their argument was, it's just software, guys, not a medical device. And FDA said, no, you're out of here. So, so it often is a medical device, and we just have to think about what kind of claims you need and what kind of functionalities in that software to classify it appropriately. But there's an awful lot of products out there that are currently labeled research use only. That as long as you're sticking to those claims, if somebody chooses to use it a different way.
I think you have to go to enormous lengths with disclaimers that it won't be used for diagnosis and reporting back to the patient or the patient's physician. I think it's a very slippery slope myself.
Yeah, I think the other piece of it is if you decide to use software as part of your process. So when we run our test, sample comes in, we do a bunch of molecular biology. All of it is tracked in a LIMS system. The LIMS system then uses an algorithm to calculate a score. The score is then translated into a likelihood of having a disease or not. And all of that software is validated through a very specific process that you need to do as part of design control, like you would with any software development product. It's no different, except you're using it, and the FDA sees that as an integral part of what you're doing. That part of your filing, even for 510K, involves the validation of your software and your LIMS system. And so it also does everything, including reporting the result back, automatically dialing the fax machine. It's very cool, actually.
That's very cool. But one thing to say, even for 510Ks, often my largest section now in 510Ks is the software validation section. So FDA has identified software functioning not as expected as a very high risk. So there's been filings where just the software validation data has been much larger than even the clinical data section.
I mean, there was one very embarrassing example. Was it 23andMe or the other guys?
Which ones?
This is where they
Flip
flipped the plate.
Yes, 23andMe.
So they had 96 samples from 96 different people that went into a 96-well plate to do a bunch of genotyping. And someone had turned the plate the wrong way. It was easy to catch because men were getting women's results and vice versa. And that was the only way. They didn't have the controls in place to catch that.
But there were some people upset. I believe one person was told that his son was not his son as a result of that.
Yeah, I didn't know.
I think there were some
Was
domestic...
John Edwards?
Domestic... No, then they would have regulated LDTs had it been John Edwards. But,
Yeah.
We're in the research stage, but we plan to go CLIA and plus or minus FDA. What's your advice or strategies for setting up a CLIA lab? I don't want to start with my garage.
you want to handle that one, Steve?
So the best advice is find some people who've done it before. That's almost always the case. I mean, California is, if you're here, is the second most rigorous state, I think, in terms of getting a CLIA lab approved. New York is first, as Maya pointed out on her slides. And I mean, there are a number of requirements. So people who work and report results in a CLIA lab need to have a clinical laboratory scientist certificate. which it turns out those people are in great demand. They get paid twice as much as people who don't have it. It's a good business to get into, generate more people with CLSs. And then, you know, remember that a CLIA license is to an address. You can only have one CLIA lab at one address. And it really behooves you to think about it as a laboratory-developed test, which is what CLIA is supposed to be. That is, the people who developed it are the people who are at least supervising the running of it. You know, getting people who have been in the regulatory, who have regulated CLIA within California, there are a number of them who are consultants now who are quite helpful in that regard. And there are people, you need a laboratory director, you need a technical supervisor, there's a pretty formulaic hierarchy of people that need to be involved specifically with it. And you can't just take your research associate who has great hands. If they don't have a CLS, they can't do anything that has to do with reporting of results. So there's a bunch of pitfalls there, but they're pretty well sorted out.
Yeah.
Maya, you want to add anything?
I think the other thing too is however you set your CLIA lab up initially, expect that you'll change it when your volume of samples changes. So often, I mean, what you hope for is that you're going to get enough stuff in that you're going to have to work on efficiency of that. So expect that there'll be process changes and lab flow changes and things like that to be able to accommodate it.
Yeah.
So whatever you do initially, assume it's going to change as you start to understand what it means to accept 100 samples a day.
Yeah, and I think the other thing is, so in California, they actually, you send in your application, and then they will come and actually do an on-site inspection and go through all of your materials and also see the lab, et cetera. And New York and California are the only states, I believe, that do that.
But some want checks.
Yeah.
So there's an extra form you have to fill out in some of the states.
And every couple of years, they come back and check again, and you have to file stuff. If you're changing the process, you have to show equivalence and on an ongoing basis and things like that. So it's an ongoing activity.
The Massachusetts companies have their CLIA labs in Maine or New Hampshire. The regulations are better and the income taxes are lower.
Yes.
So FDA, they're going to get rid of LDTs? Or resident?
That's the million-dollar question. FDA has written all the guidance documents. If you go,
Can you repeat the question, please?
I'm sorry. The question is, will FDA ever regulate LDTs? If you talk to FDA, so there was a roundtable meeting where the deputy director, Don St. Pierre, was speaking at 2 weeks ago. Somebody asked him about LDTs, and his response was, I would be willing to give blood to get these guidance documents through. Apparently, the people at the Office of Management and Budget don't want my blood. So as far as see, if you read my blog, Steve, you would have seen that already. But so FDA.
I have the link to your blog.
We'll work on
But
that.
I have 3,000 emails in my inbox, so it probably got lost.
All right, okay. But anyway, but if you ask the folks at FDA, they definitely are very serious. I think with the T21 tests that have come along, I think Alberto Gutierrez is particularly worried about the T21 testing of the fetal cells and the maternal cells. I think they're very worried about the risk of that. They're very worried about the risk of particularly chemosensitivity assays. So if you ask FDA, they will give you the statement that they believe it's important to regulate these and that they would put this into place over a period of, usually the guess is 5 to 7 years, but there appears to be some point of stoppage within the White House, and they have not been given authority to do that. So I actually lost a bet to Steve at some point about this because
You
I
still
thought
owe me something for
I
that.
do. I owe you lunch, but I stopped making bets on it because this appears to be a pretty big policy change that FDA has not been successful in getting through.
Yeah.
So
Just to be clear, I mean, this has been 3 or 4 years where we
more
thought
than
guidance was going to be finalized, it was going to be done, there was a comment period, then it sort of disappeared into a black hole, then it came out as we're managing risk, and instead of being what was called an IVD-MIA program, now it was an LDT program, and so, I mean, there are legitimate issues around regulation versus innovation and what's the right balance between those. And I think that's what people are struggling with. Plus the issue is if you keep cutting FDA budget, how do you expect them to regulate more stuff, as well as the food supply, as well as everything else they're being asked to do on less money?
right.
And that won't work well.
But what I do advise in order to handle this risk, and if you'd eventually see FDA in your future, is please put in some kind of design control, some kind of development control process. I think it will be valuable whether you go through FDA or not, and that's something you can't reproduce later.
Yes.
Sort of kind of magically coming up with a design history record later is pretty tough.
It's called fraud.
That too. The other thing is that the one thing that CLIA does not cover software validation. So if you are heavy on software, you are doing an algorithm approach, I would suggest looking at the FDA guidance documents around software validation. Again, I don't think it's just paperwork. I think it's something that will be valuable for you as you develop and change that software to keep track of changes, things like that. And then CLIA also doesn't require auditing of your suppliers. And so that may be a program that you want to put in place, and there's some tricky issues there, but that may be something you want to consider. Again, I don't think it's just paperwork. I think you can learn a lot by actually going out and seeing how some of these RUO products are made. It may be actually illuminating for you.
I think there's one other thing. I'm surprised you didn't bring it up.
I don't know yet.
Which is that especially if you're getting ready to set up a large prospective trial where you're going to spend a lot of money, go talk to the FDA, have a pre-IDE meeting. It doesn't force you to file, but get at least their buy-in around two things. That the study design is appropriate for the question you're trying to inform upon, and that the intended use statement of who the patient population is makes sense based on the trial, or doesn't make sense based on the trial design. Those two questions are really key, and FDA is perfectly willing. You don't have to, it isn't a huge amount of work to prepare a pre-ID document. It's no more than a dozen pages. And even before you have a product, it may be worthwhile doing that, because, I mean, we did this before we started our initial large prospective study to say, okay, here's the study design. Do you agree that this is the right study design to answer this question? We recognize there are all these issues, but do you agree? They did agree. They may change their mind later, and we subsequently ran another study to overcome what we thought was the major objection, and we thought it was too. But that's worth very much contemplating, because at the end of the day, they will give you good advice. You may not want to hear it, and you can decide today in this environment whether you need to follow it or not. But it is useful information, and it really doesn't cost you anything.
So one last question, if there is one.
So we talked about existing and prospective cohorts. So we talked about time considerations, patient populations. What are some of the cost considerations for that decision?
So the question is around existing cohorts, what the cost considerations are of buying a cohort, or?
Yeah, so well, making a decision on whether to go with an existing cohort versus a prospective cohort. Right?
If you can afford it, always do the prospective one. I'm serious.
What's the delta around making that decision?
Let me give you some idea. I spent $2.8 million for 3 times patient.
So think about it this way. To get a blood sample as part of a prospective trial probably costs $1,000, round numbers. Maybe it's 2,000, maybe it's 800. It's not a lot less than that. So if you need to run a 1,000 patient study, you do the math. Okay. Now, you can get, and by the way, all the universities and cooperative groups who have samples know this math too. So you're not going to get things a lot cheaper. What you save is potentially is time. And time is extremely valuable. If you can get on the market 2 years sooner, that's worth a lot. The challenge you have is the one that was brought up earlier and reiterated here. Almost certainly, the samples weren't accrued exactly the way you've developed your test. That turns out, I'll give you a very simple example. If you are collecting blood samples and you draw the blood into a syringe first, and then put it into a tube for preservation versus doing a standard venipuncture where it flows directly into the tube, you completely change the gene expression patterns of the cells that are in that blood. Because you have shear forces, you get degranulation of neutrophils, completely different. If we hadn't recognized this when we were developing our test, we would never have been able to develop it. Because we had an initial cohort that had been done with the syringes, and when we went to our trial, we realized before we started it that you couldn't do that. So, I mean, there are subtleties like that that I mean, there was just a paper, I don't know how many of you saw, there was a paper that got a lot of press recently from Eric Topol in Science Translational Medicine about circulating endothelial cells and likelihood of heart attacks. And one of the things, if you read the fine print, was that their control patients who did not go on to have MIs were drawn by normal venipuncture. And the patients who were then on their way to the cath lab, A, had been treated with heparin, and B, it had been done with a syringe. And I know Eric because he was our PI on one of our studies, and I emailed him and I said, Eric, you know about this? He said, yeah, we did the controls. It's okay. But how you handle the blood samples is very important. So there are these little details, not so much with genetics. With DNA, you can do just about anything with DNA. But when it comes to anything else, RNA or proteins or metabolites, our bodies are made to respond to
this is an area where the FDA can almost be a hindrance. In the US, we have to use stored bloods until it's approved. You know, we can't do our actual fresh tests that we can do in other countries. If you store blood and the red cells start to break down and there are all sorts of secondary metabolites of things in blood that really confound the tests, yet until it's approved, you can't go in and do finger sticks, which is what we do in every other country in the world.
And I will say, I agree with Steve on the oncology samples. You know, that sometimes you need those outcomes, so you have to go there. But I would be wary. FFPE varies significantly by institution, and I've seen more than one company stopped by understanding if their assay can handle the variation in how those specimens are preserved. And there are just certain technologies that don't cope with FFPE at all well. Plus, those blocks will vary. So you may get a section that has a lot of tumor cells in it, something like that. So I absolutely agree with him that those are the kinds of samples that we typically go after retrospectively, but it's not straightforward.
Yeah. You always have to do the multivariate analysis, say what's the determinant of disease? Is it the center where it was picked, or is it actually something you're measuring?
Yeah.
And you always have to do that.
Yes, and people try to, you know, we're just going to get this slices or something like that. We're not going to get the whole block, and they'll just be, and do you have to do punches? Do you have to, there's all kinds of issues with it. So I agree, if you're in oncology, you may want to go that direction, but it won't be straightforward.
Thank you so much for your time. Really appreciate it. Thank you.
We didn't even fight.